Imagine a world where limes taste like oranges, or where cats chirp like birds. Sounds strange, right? But this isn't just the stuff of science fiction. In the world of genetics, such fantastical combinations aren't so far-fetched. Scientists are constantly pushing the boundaries of what we know about DNA and genetic engineering. This is the world of hybrid DNA, where the building blocks of life are mixed and matched to create something entirely new. By combining the genetic material of different species, researchers can produce organisms with unique traits and characteristics. Hybrids, as we often call them, are all around us. They can be found in our grocery stores, our gardens, and even in the wild. From the tangy sweetness of a grapefruit, a hybrid of a pomelo and an orange, to the vibrant colors of ornamental plants bred for their beauty, to the majestic beauty of a liger, a hybrid of a lion and a tiger, hybrids are a testament to the creativity and ingenuity of nature and science. Hybrid organisms challenge our understanding of what defines a species. They blur the lines between distinct categories, showing us that life is more fluid and interconnected than we might have thought. They demonstrate the incredible power of genetics to shape life as we know it. Through careful manipulation of DNA, scientists can create organisms that are stronger, more resilient, or have entirely new abilities. But what exactly is hybrid DNA, and how is it created? The process involves isolating and combining specific genes from different species to produce desired traits. Let's delve into the fascinating world of genetics to find out. Join us as we explore the science behind hybrid DNA and the incredible possibilities it holds for the future. DNA, or deoxyribonucleic acid, is the blueprint of life. It carries the genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms and many viruses. It's a long, winding molecule found in the nucleus of every cell in our bodies. This molecule is composed of two strands that coil around each other to form a double helix, carrying the genetic instructions. Think of DNA as a massive library filled with books containing instructions for building and maintaining a living organism. Each book in this library is a gene, and each gene carries a specific set of instructions. These instructions are written in a code made up of 4 chemical bases: adenine, A, thymine, T, cytosine, C, and guanine, G. The sequence of these bases determines the information available for building and maintaining an organism. Hybrid DNA is created when segments of DNA from different sources are combined. This can happen naturally or be engineered in a lab. Imagine taking a chapter from a book about a lion and inserting it into a book about a tiger. The result is a new book with instructions for a creature that possesses traits from both animals. This new book, or hybrid DNA, can create an organism with characteristics from both parent species, like a liger, which is a hybrid of a lion and a tiger. This mixing of genetic material can happen naturally, as seen in the case of mules, hybrids of horses and donkeys. Mules inherit traits from both parents, making them strong and resilient. Alternatively, hybrid DNA can be achieved artificially in a laboratory setting. Scientists can manipulate DNA to create new combinations, leading to advancements in medicine, agriculture, and biotechnology. Scientists have developed sophisticated tools to cut and paste DNA segments with incredible precision. Using enzymes that act like molecular scissors, they can snip out specific genes from one organism and insert them into the DNA of another. This process, known as genetic engineering, allows scientists to create hybrid DNA with desired traits. Imagine wanting to create a tomato plant that is resistant to a specific disease. Scientists can identify the gene responsible for disease resistance in another plant species and insert it into the tomato's DNA. The resulting hybrid tomato plant will inherit the beneficial trait, protecting it from the disease. This ability to manipulate DNA has revolutionized fields like agriculture and medicine, offering solutions to some of the world's most pressing problems. One fascinating aspect of hybrid DNA is the phenomenon of hybrid vigor, also known as heterosis. This occurs when hybrid offspring exhibit superior traits compared to their parents. For instance, a hybrid plant might be taller, produce more fruit, or be more resistant to pests and diseases than either of its parent plants. This enhanced performance is attributed to the increased genetic diversity within the hybrid. By combining genes from different sources, the hybrid inherits a wider range of alleles, variations of a gene, which can lead to a more robust and adaptable organism. Hybrid vigor is a testament to the benefits of genetic diversity and highlights the importance of preserving biodiversity in nature. Section 5. Hybrid DNA and humanity. Are we all hybrids? The concept of hybrid DNA might seem like something straight out of a science fiction novel, but it's more relevant to our own species than we might think. Hybrid DNA refers to the genetic material that results from the interbreeding of different species or subspecies. In the context of human evolution, this means that our ancestors mated with other hominid species, leading to the genetic diversity we see today. Human evolution has been shaped by interbreeding with other hominid species, such as Neanderthals and Denisovans. These interactions were not just rare occurrences, but were significant enough to leave a lasting impact on our genetic makeup. Fossil evidence and genetic studies have shown that these ancient humans coexisted and interbred with our direct ancestors. Studies have shown that modern humans carry traces of Neanderthal and Denisovan DNA in our genomes, indicating that our ancestors interbred with these now extinct hominids. This genetic legacy is not just a historical curiosity, it has practical implications for our health and biology today. For instance, certain genes inherited from Neanderthals are known to influence our immune response and susceptibility to diseases. This interbreeding has introduced new genes into the human gene pool, influencing traits such as our immune system and our adaptation to different environments. These genetic contributions have helped humans survive and thrive in diverse climates and conditions, from the frigid Arctic to the sweltering tropics. The ability to adapt to various environments is a testament to the resilience and versatility of our species. The discovery of these ancient gene flows challenges the traditional view of human evolution as a linear progression and highlights the interconnectedness of all life on Earth. It suggests that human evolution is more like a web of interactions and exchanges, rather than a straight line. This interconnectedness underscores the idea that we are all part of a larger tapestry of life, sharing a common heritage that transcends time and space. Section 6. Ethical Considerations, a Brave New World of Genetics. The ability to manipulate DNA raises profound ethical questions. While hybrid DNA technology holds immense potential for good, it also presents risks that must be carefully considered. For example, the creation of genetically modified organisms, or GMOs, has sparked debate about potential environmental impacts and the safety of consuming genetically modified foods. Furthermore, the ability to alter the human germline, the DNA passed down to future generations, raises ethical concerns about designer babies and the potential for exacerbating social inequalities. It's crucial to establish ethical guidelines and regulations to ensure the responsible development and use of hybrid DNA technology. Open and informed public discourse is essential to navigate these complex issues and ensure that scientific advancements benefit all of humanity. Section 7. Hybrid DNA in Action. Real-world examples. Hybrid DNA technology has already made significant contributions in various fields. In medicine, for example, scientists are developing gene therapies that use modified viruses to deliver therapeutic genes into patients' cells, offering potential cures for genetic diseases. In agriculture, genetically modified crops have been engineered to resist pests, tolerate herbicides, and improve nutritional content, leading to increased food production and reduced reliance on pesticides. Hybrid DNA technology is also being used in bioremediation, where genetically engineered microorganisms are deployed to clean up pollutants and restore contaminated environments. These examples demonstrate the transformative potential of hybrid DNA technology to address some of the world's most pressing challenges. Section 8. The future of hybrids. What lies ahead? As our understanding of genetics continues to advance, so too will our ability to manipulate DNA. The future of hybrid DNA is filled with both promise and uncertainty. We can expect to see even more innovative applications of this technology in medicine, agriculture, and beyond. However, it's crucial to proceed with caution and ensure that ethical considerations remain at the forefront of scientific progress. Open dialogue, public engagement, and responsible regulation are essential to harness the full potential of hybrid DNA technology while mitigating potential risks. The future of hybrids is ultimately in our hands. It's up to us to shape a future where this powerful technology is used responsibly for the betterment of humanity and the planet. Section 9. A final thought. The study of hybrid DNA reveals that the boundaries between species are not as clear-cut as we once thought. It challenges our understanding of what it means to be human and highlights the interconnectedness of all life on Earth. As we continue to unravel the mysteries of genetics, one thing remains certain: the future of life on Earth will be shaped by the choices we make today.